Okay, it looks like we have almost everyone, so I'll get started. Today, what we're going to focus on is the concept of genome annotation, what genome annotations are, how they're derived both experimentally and molecularly, and computationally. And I'm also going to walk through, in the second part of the lecture, some of the more common and very useful resources that are available to get genome annotations into your analyses. And the basic motivation behind all this is it's one thing to sequence a genome, a reference genome for human or for mouse or for fruit fly. But it is very difficult to do anything in terms of functional analysis or interpreting genetic variation or figuring out genes or loci that might be associated with a trait without knowing, first of all, where genes are in that FASTA file that you get from a reference genome, where genetic variation is and isn't, which parts of the genome are conserved across species, which seem to be specific to a particular lineage, for instance. And by and large, those types of annotations come from computational predictions. So I'm going to focus mainly on the computational aspects because that's really the focus of this course, but certainly many of these annotations are a combination of molecular and computational approaches. So just big picture for the next couple lectures. Today, we're going to talk about what genome annotations are and where we can get genome annotations for using them in our research. The next lecture is going to be talking about some of the formats that we use for genome annotation, going into a bit more detail there. And then we're going to have a lecture, perhaps two, I haven't officially decided yet, on using a piece of software that I wrote a long time ago and that my lab maintains called Bed Tools. It's for using genome annotations in BED and GFF format to analyze and interpret your experimental data in the context of these genome annotations. Okay, so just a quick heads up, I'm going to assign homework 6 sometime this week. I'm still refactoring things a little bit. I'm thinking probably assign it for Wednesday or Thursday, and we'll have a week to finish that up. But I'll announce that through email and Slack. Okay, so genome annotation is motivated by the fact that when we sequence a reference genome for any species, we get a big FASTA file that basically looks like this, but for page after page after page after page. So that's useful. It's obviously nice to know the genome sequence, but we really need to be able to know which parts of this sequence encode genes, which regions are intergenic, which sequences might be regulatory elements or motifs that a particular transcription factor binds to, which sequences are restriction enzyme cut sites, which parts of this genome are enriched for meiotic crossover events, where's genetic variation. So we really want to be able to go through and essentially color code or annotate, highlight different regions of the genome. And that is really the whole concept and field of genome annotation. I think in many ways we take this for granted, especially in human genomics. There's just in mouse, for instance, probably all more model organisms, commonly used model organisms. The reference genomes have been around for 20 years. They're really well studied organisms, so there's just a lot known. And it's we can go to things like FlyBase if you work on fly, or I think it's called WormBase if you work on nematode. Human, there's multiple different websites that you can go to figure out what are all the disease genes, where are CPG islands, where's all the genetic variation. But if you're studying an emerging organism or trying to sequence some new critter, which is increasingly common now given the the accuracy and the throughput of sequencing technologies, you actually start at the very beginning. I mean, you first have to collect DNA from that new organism and put together a genome, which is a complicated task in and of itself. But once that's done, you then have to figure out, you have to annotate that genome to do anything useful with it. And that, in some ways, is a straightforward process. Some aspects of that are straightforward, but there are many aspects of it that are actually quite difficult and require often years of research just to figure out essentially where are the landmarks in this genome. I think it's very much like, we take Google Maps for granted, that we can just type something in and know where to go. Well, that's what genome annotations give us for genomics. So let's imagine we're sequencing, what's this guy's name? Gary? I think that's Gary from SpongeBob the Snail. If I got that wrong, let me know. It's been a while since I watched the show. But imagine we were able to get DNA from Gary, and we are able to see, we got funding from either our university or our collaborator or NIH or NSF or something, probably NSF for Gary. And we sequence his genome. We spend a couple months figuring out how to do the assembly correctly, and now we want to annotate it. And I'd say the first step for annotating Gary's genome, or really any critter's genome, is to figure out where the genes are, right? I mean, that's the business end of any species genome is the protein coding genes, right? That's most of the phenotypes that we're interested in. Maybe we're studying Gary because he's got these beautiful big eyes. What genes drive those huge eyes that Gary has, and why are they orange? So to do that, we can do a couple of things. There's ways to figure out where genes are molecularly. So what if we had a reference genome, what molecular experiment, what sequencing experiment could we do to maybe guess as to where the genes are? Anyone have any ideas? What could we do? No computation. We would just isolate some molecule from Gary's cells and maybe have some guesses as to where genes might be. Anyone have an idea?
Some kind of like DNA footprinting, where you look at where the polymerase is binding on the genome.
Yeah, we could definitely do that. There's what, so you went with DNA, so that would work. What's another molecule that might be useful for figuring out where genes might be?
RNA.
RNA, yes. So we could just say isolate RNA from Gary's eyeballs and convert it to cDNA and map it back to this reference genome that we have, right? So when we align it back to the reference genome, that's going to give us a pretty strong hint as to where exons are, right? If we have mature RNAs that we converted to cDNA, that's a direct way of predicting where exons might be. And we can infer that when we align to the exons, that everything in between is introns. Another thing we could do to riff on what I think Jordan mentioned is we could look at regions where chromatin is open or accessible. And so we could do things like DNase 1 hypersensitivity assays, or the modern equivalent of it, his name is escaping me. If anyone remembers, let me know. But there's all sorts of assays now where you can use tricks to figure out what are open regions of chromatin, which tend to be expressed, or genes nearby tend to be expressed, and closed regions tend to be heterochromatin with little expression. So those are molecular approaches, but that costs more money. One aspect of genome annotation is the fact that we can actually leverage existing databases to make some of these predictions. So let's imagine that there's some other snail that is a close relative to Gary that has already been sequenced and annotated. we can use the notion of evolution essentially to identify known sequences or annotations in a closely related species to find homologous sequence in Gary's genome and make the inference that these, because they're similar to this other species, these parts of the genome might encode genes or have similar function as in the other species. And so that computational prediction is going to have some degree of accuracy. If it's a very closely related species, let's say most of those predictions will be right. 90% of our gene predictions based upon homology may be right, just a guess. But we would still want to then pursue experiments to make sure that those computational predictions actually bear out. So for instance, if we predict 25,000 genes in Gary's genome, we might want to use RNA sequencing, for instance, to figure out which of those gene predictions actually exhibit expression. There are transcripts observed for those sequences. And if we do multiple replicates and we find from multiple tissues in Gary's, from Gary, we might find that some of those 25,000 gene predictions never have any gene transcript expression. So we might curate our annotations to say, well, maybe these actually aren't protein coding genes in Gary's genome. But using those annotations, that's where we can start designing downstream experiments to try and map traits in whatever species Gary is. But it all depends upon this iterative and rather important and complex process of genome annotation. So very high level, we saw this slide last lecture. Annotation really provides context. Without this annotation, we just have essentially naked FASTA files, naked genomes that are, unless you're a savant, really impossible to do anything with. You can't just scan through, page through a big FASTA file and go, oh yeah, there's a gene, there's an intron, there's a genetic variant, there's a PFAM domain. We depend upon these databases. So let's say that we spent all the money that we had sequencing Gary's genome. We collected DNA and we were able to get some PacBio sequences and some Illumina sequences, and we assembled this glorious genome. And now we don't have any money left to predict where the genes are molecularly. We can't do that RNA-seq experiment. We can't do that DNA footprinting experiment. So we want to annotate genes computationally. How do we go about doing that? Does anyone have any ideas as to, like, if we, just based upon what you've been taught in molecular biology and the central dogma of molecular biology, what are some tricks that we could use to guess as to where genes might be in a huge FASTA file for Gary's genome? If we were to write a piece of software to do this, any tricks that we could think of? What's something we know about gene structure?
You could look for the start and stop codons and the nucleotides that encode them.
Exactly. So start codon is what, AUG, right? So you could look for that. You could look for downstream stop codons. So that's a first pass. You could basically just go through the entire genome and computationally highlight where potential start and stop codons are. Yeah, absolutely. Now that will give you a nice first, I'd say that's like a rough pass. Then you need to do another pass through the genome to do some more analysis, because, you know, AUG is just a triplet, and the probability of seeing, well, ATG, I guess, or its complement in the genome is pretty high, just randomly. Looks like a suggestion in chat here, maybe. Yes, so looking for Tata boxes, yep. for transcription, factor binding, and initiation. We could certainly do that. What about introns and exons? What do we know about, let's say we're dealing with a eukaryote. What do we know about splicing of introns and splicing out of introns in genes?
We know a lot of the splicing site binding sequences.
Right, exactly. So we know, for instance, we know canonical donor acceptor sites at exon intron boundaries. We also know that a lot of introns have polypyrimidine tracks, which has to do with splicing machinery binding and recognizing those motifs. A couple other things building upon that. Those are all great suggestions. Thank you. We do know that coding sequence tends to be more GC rich than non-coding sequence. So we could sort of scan through the genome and look for higher GC regions. But I think, Jason's suggestion, Holly's suggestion, and I maybe lost one other suggestion, sorry, Avitra maybe. We could combine all those are each independent signals really, and that they each can be combined to start honing in on where coding sequences are and are not. So that would be the first pass. But then the beauty is that we can also use annotated genes from other species. There's this notion of shared evolution. And so we could, if there is an annotated species that's nearby, well, let's align Gary's genome to some other, I don't even know the name of the Latin name of a snail species, but let's imagine there is one, we can align them, we figure out where the sequences are similar, and then we can essentially map the annotations that exist in this other species genome to Gary's genome to start honing in on where gene annotations or gene sequences might actually be. So we covered a lot of these, start codons, donor acceptor sites we can look for. The other thing we know is introns tend to be big, exons tend to be relatively small. The average exon size in the human genome is about 150 base pairs. So if we start seeing acceptor and donor sites, on average 150 base pairs away, then we might believe it. If we see an acceptor site and a donor site 5 kb away, we would be a little more skeptical that interstitial 5 kilobase sequence is an exon, given that our prior, going back to Bayesian expectation, our prior expectation is 150 nucleotides. Now, obviously that doesn't work. There are huge exons and there are small exons, but so essentially we can use what we know about average intron size, average exon size, and these sequences that should be flanking all these to help. But certainly what has expedited this process the most is using homology between similar genomes of similar species to essentially project annotations that already exist in one species onto a similar genome from a similar species. Right, and then we already covered this. We can then vet those predictions if we then, okay, so we spent all our money getting the genome, we've mapped over annotations from another species, and we've done all we can for free. Now we write the grant or the fellowship application, which says, all right, well, we'd like to do some RNA sequencing, and we'd like to do some DNA footprinting, and we'd like to do whatever the modern version of DNA's hypersensitivity sequencing is. If anyone remembers, it's driving me nuts, let me know what it is, I forget. We could sequence those RNA molecules with Illumina sequencing, and remember that since we've sequenced mature RNAs and converted it to cDNA, when we align those sequences back, we're going to have nucleotide resolution mapping of where introns and exons are, because the cDNA just contains exons. There's going to be part of that cDNA molecule that lines to, say, 1 5' exon, and that's going to skip the intron and align to the 3' exon. And so we can infer that everything between we can infer two things. Everything in between is an intron, at least in one isoform, and that annotation, that prediction that we gathered computationally is vetted. There actually is messenger RNA expressed from that prediction. So this molecular pass can help us refine those annotations. And we can start doing things in a tissue specific manner. We might be able to figure out which genes are expressed in one cell type and another cell type. Okay, so what I'm going to do now is walk through some other types of annotations that we have access to for the human genome and for really any genome. There's a collection of software tools that are used routinely to go beyond just annotating gene sequences. So actually, I should mention that if you are doing this, if you're working in a new species, or that might be something that's going on in your lab, Mark Andel's group here has a really fantastic piece of software called Maker, which basically goes through the thought experiment that we just did, where it takes closely related species and a lot of other information to take a naked FASTA file and start to figure out where gene sequences might be. But then the next step is using other tools to find, for instance, repetitive sequences in the genome. So we know that many eukaryotic species, especially vertebrates, their genomes are littered with tons of repetitive sequences. And those include things like on the bottom here, which are these simple sequence repeats. So for instance, this row in this screenshot from the UCSC genome browser shows instances of the repeat, looks like 8 A's followed by a G followed by 2 A's. And that annotation exists right here in some part of this chromosome that we're looking at. So there's lots of these. You can see that these are repetitive sequences because it's, repeats of A, T, or higher level repeats that are harder to see. We also know that there's repeats from retrotransposition. So that's these lines and sign elements, DNA repeats, microsatellites, all these repetitive sequences. And the way that these are found are with two tools. One is called TRF, or Tandem Repeat Finder, and that's literally the name of this track. It's called Simple Tandem Repeats by this tool called Tandem Repeat Finder, and it finds all these sequences by looking at the FASTA file. Now, anyone have a guess as to how it does that? How does it find repeats? What is it actually looking at? If it wanted to find, for instance, if it wanted to find all of these repeats, first of all, it has to know what repeats to look for in a particular genome. And there's a database called RepBase, which has a catalog of all known repetitive sequences. So in its crudest form, what you could do for repeat annotation is, for each repeat type that's in this database, run grep on the FASTA file and mark each of the instances of that exact match of that repeat. Now that's not the best approach, but conceptually it would kind of work. It's not the best approach because you might have subtle differences between the repeat that you're looking for and what's in the genome that you're testing. So for instance, instead of this 8 A's followed by a G followed by 2 A's, Gary's genome might have 8 A's followed by GG followed by AA. Would we want to say that that's not a repeat? Well, it is a repeat. It might be a new repeat type that's in Gary's genome. So essentially, these databases can be used to have a crude estimate of what repeats might be in a particular genome, but the tools like Tandem Repeat Finder and another one called Repeat Masker are allowing for subtle differences between what's in the database and what's observed in the FASTA file of a particular species. And at the end of the day, what it ends up doing is just, like highlighting or marking the coordinates or the positions in the FASTA file at which a particular repeat was known or observed, and then it marks what repeat type it was. So that's how we get these nice visualizations in something like UCSC Genome Browser. Where are all the signed elements? Where are all the line elements? Where's all the repeats of this type or this type or this type? Where's all the DNA transposons? That all comes from computational predictions. This is not done molecularly at all. RepBase and Repeat Masker and Tandem Repeat Finder are run on essentially every single new genome that is sequenced and submitted to resources like UCSC. Okay, so another thing that we may want to do is track GC content, or to actually measure the GC content along every single chromosome. And if you've ever designed PCR primers or guide RNAs for CRISPR-Cas9 experiments, you know that GC content matters. I haven't done PCR in 15 years, but my memory is, when you use, I don't even remember the name of the tool, the primer design tool, it will sometimes reject candidate primers because of excessively high or excessively low GC content, because they may not behave as you expect, or you want the two primers that you use to have similar behavior so you get consistent amplification. So how do we go about measuring GC content? Anyone have a suggestion for how we might, let's imagine we have a program. We're not writing any code here. We're just thinking about what the code might do. It opens up the FASTA file for Gary's genome and starts at the first nucleotide of chromosome 1 for Gary. How might we measure GC content along chromosome 1 for Gary's genome? Any ideas? What do we have to know to measure GC content?
The number of G's and C's compared to the total number of nucleotides.
Yes, absolutely. And so the first decision, so we have to count things. We have to count how many G's and C's we observe in Gary's genome with respect to a total. How do we define the total? Well, the total could be done in two ways. One, you could just measure the total number of G's and C's out of all the nucleotides on chromosome 1 for Gary's genome. So that will give you a chromosome-wide GC content measure. But as is depicted in this figure, GC content varies dramatically along a chromosome. So if we were to use GC content as a proxy for, say, gene sequences, so the higher the GC content, the more likely we believe that a gene body might exist within a particular region. We need to do something more sophisticated. We have to break down chromosome 1 into smaller chunks and measure the GC content in each of those chunks. So in this plot, we're just looking at a relatively small region of chromosome 19 from the human genome. And each of we have this oscillating red line that reflects the observed GC content. Let's just say it's for a 500 base pair window. Each point along this fluctuating line is a 500 nucleotide window. So that means, say, from position 1 to position 500 on chromosome 1 for Gary, if we're doing the same thing, we count how many G's and C's we observed out of the 500 nucleotides that were in that window, and that's our GC content. So if it was 250 G's and C's and there's 500 nucleotides, the GC content is 0.5. We step forward one nucleotide and we now measure from position 2 to 501 and move along. So we just keep marching along and measuring the GC content. So it's a fairly simple process, but it's rather computationally laborious. There's a lot of measurements to take, but it's super useful for, very, very critical annotations, like where are genes, where are not genes. So for instance, this region right here with GC content of 0.35 is pretty unlikely to contain gene sequences, whereas sequences, regions of the genome that are up here around between 0.45 and 0.55 GC content are more likely to harbor genes. Okay, so that's another computational prediction. It's a gimme. We don't need to do molecular experiments. Basically, as soon as you have your FASTA file, you run a script. I don't know if Maker has a script for doing this, but if you wanted to challenge yourself with a little coding experiment, you could probably, if you know any R or Python or Perl, or wanted to learn one of those languages, a nice first experiment might be to just measure GC content along a FASTA file. Okay, so another thing that we touched upon early on, but I've ignored for a little bit, is this notion of conservation. In order to understand what nucleotides or amino acid residues in a particular species might be relevant to its fitness or particular traits, we often want to use the fact that we've sequenced genomes from other closely and distantly related species to figure out what residues are the most conserved. And the inference is that amino acid residues or even nucleotide regions that are highly conserved, not only across between Gary's genome and similarly related species, but also maybe back to yeast or E. coli or in other vertebrates. If they are highly conserved, the inference is that they're probably important. They have something to do with essential function in that species. So I think this is a pretty clear concept, but essentially what you're doing is you align the genomes or amino acid sequences in this case. So we're looking at amino acid sequences. I've forgotten what protein this is, but between human, yeast, and Plasmodium. And you could see that for whatever protein this is, there are some residues that are conserved from human all the way down to Plasmodium falciparum, which, that really speaks to this notion of shared evolution, and these residues that are conserved across all these distant related species are probably relevant to either the structure of the protein or the active sites within the protein. I don't know what this is. Maybe it's a kinase, maybe it's a structural protein, we don't know. But the more conserved across species, the more likely to be functional. You've probably, if you've never been to the Natural History Museum up the hill, go. I haven't been in 2 years, but at least 2 years ago, there was a really fantastic exhibit up on the top floor of an amino acid sequence alignment like this, but for keratin, and showing the conservation of keratin and its use in many different things across species, everything from hair and skin and other things. And it's just a very cool, clear example of conservation and how it demonstrates shared function across these distant related species. So this is another thing, a phylogenetic conservation measurement through multiple sequence alignment or of either genomes or amino acid sequences is a common step that is done for every new genome species. In human and other vertebrates, we tend to use measurements like FastCons and GERP and PhyloP. These are tools written by legends in that field to do this very accurately and computationally efficiently. And we take these for granted, but essentially there's tools that are being run on all these genomes to come up with these conservation measurements. Okay, oops. So let's zoom in on this. What we're looking at now up top is a little snippet of chromosome 2 on what's known as build 19 strangely also known as build 37 of the human genome. I'll come back to that in a little bit. But what we're seeing on the x-axis here is coordinates along this part of chromosome 2, ranging from position 30.453 million to 30.455 million. And on the y-axis is the degree of conservation at the nucleotide level, not at the amino acid level, across mammals that have been sequenced. So you can see that some of these regions there's very little conservation across all the species, but all of a sudden we hit here, and it's even at the nucleotide level is hugely conserved across all the species that were measured. And what you can see as we add in annotations for genes, so the convention in the UCSC genome browser is that protein coding exons are these thick rectangles. Untranslated or non-coding exons, or UTRs, are these thin rectangles. And then a thin line is used to represent an intron, and the arrows are used to denote the direction of transcription, the strand of transcription. The point of showing this is that we know where we can also predict what the start of translation is. This is part of the annotations. And what you can see is there's not much conservation 5' of the gene body. But when we get into the gene body, the conservation across these species really ramps up. Many more of these nucleotides are conserved. And I think it makes sense that we see high conservation right around this start of translation. And then when we zoom in on this little window right here, right around the start of translation, we get to see all of the sequences from mammals that were aligned here. And we can see not only the conservation at the nucleotide level, but then what's so amazing about the UCSC genome browser is when you zoom in at this level, not only do you automatically see the conservation across all these species at the nucleotide level, but also at the amino acid level in all 6 potential reading frames. It's a pretty incredible resource. So what we can see is that this, right in this region, we have high conservation. The basic point is that it's conserved almost identical residues across all these different species, ranging from elephant to human to armadillo to something called a microbat and a megabat. I actually don't know what those are. If you do, holler. But I think what's really powerful about this is you can directly, either with your own programs or using something like the UCSC genome browser, go in for any gene that you're interested in and inspect the degree of conservation across all these species. Okay, so all these annotations, we've talked through molecular and computational approaches to take a FASTA file and predict where and where not different types of annotations are. One way that we could store, oh, thank you, Michelle tells me what microbat and megabat. Oh, really? Small and big? Microbat and megabat, geez, okay. Well, aren't there like thousands of bat species? So I imagine that got old pretty quickly. Anyway, so one way of annotating or storing all these annotations for, say, GC content or for genes or for repeats of a given type would be to define the sequence itself. What is the sequence in Gary's genome? What are all the sequences that are sign elements? Well, the problem with that is you still don't know where those sequences are in the genome. You would have to take all those annotations and then align them to the reference genome to figure out where they belong. So instead, a much more clever way is to use the genome, the reference genome, as a coordinate system. And what I mean by that is if we know the length of every chromosome, so let's say chromosome 1 in Gary's genome is, let's keep it simple, 100 million nucleotides long. That means that the FASTA file starts with the first nucleotide and ends with the 100 millionth nucleotide. But what we can use is just like GPS coordinates for defining, unambiguously defining coordinates on the globe. We can use a chromosome, a start coordinate, and an end coordinate to define exactly where a particular annotation belongs in Gary's genome. So if we know that the second exon of a particular gene is on chromosome 1 at position 5 million to 5,100,000, we don't have to care about the nucleotide sequence anymore. We can just, the shorthand for the location of that annotation or that exon is just chromosome start end coordinate. And then if we want to know the sequence, we can just jump to that appropriate coordinate on chromosome 1 and actually look at what the nucleotide sequence is. So I think it's so conceptually simple and obvious now, but actually when all this genome sequencing work was being done, it wasn't so obvious. And it's a fantastic way to navigate and compare and contrast and find things in the genome, but we sort of take it for granted now. So concretely, here's a little snippet of chromosome 3. Just made this up, and we can define. All right, well, there's an annotation from here to here, so we can just report in a file. We can say on chromosome 3, from position 16 to 20. That is some motif that we care about, and we can in a file just put chromosome 3, 16, 20. And then some description of that, and it's good forever. And you can use things like the UCSC genome browser to then jump to that location and look at the nucleotide sequence. So all we have to store is the coordinates rather than the nucleotide sequence. And so this is what's done. Well, when you look at something like the UCSC genome browser and you see all those nice lines and exons and introns and all those graphics on the browser, behind the scenes, all that information is stored as these 3 key pieces of information: chromosome, start coordinate, end coordinate. And then in addition, there's a description. There's from position 5 to 10, gene A is there, or maybe some other cool, you can use this to annotate some interesting or cool locus that you care about, or maybe this region led to some important discovery and you can annotate it that way. So it's incredibly flexible, is the point. In the UCSC genome browser and other databases like it, they have database tables, or conceptually spreadsheets, of just all these different intervals in a genome on this coordinate system that define where particular feature or annotation types are. Okay, so what I want to focus on, let's see how much time I have, great. What I want to focus on over the last 20 minutes or so, 20 or 30 minutes, is to show you how to go about finding and downloading genome annotations. So just, I forget how you do this, but you probably know. If you could raise your hand, the little icon thing, and tell me if you've ever downloaded annotations from Ensemble or the UCSC genome browser, just let me know. Excellent. All right. Well, Jordan and Jason and John may be a little bit bored. I apologize. I'll try to keep it fresh for you, but I think the rest of you will learn something super useful. Okay, so the aesthetics of the UCSC genome browser are certainly out of the early 1990s. It's not the prettiest website in the world, but it's incredibly useful. So what we're going to do, if you could just follow along with me, you can watch the slides, but I think actually if you can watch the slides, but maybe do this on your own genome browser, it'll help probably cement the idea. It's the genome browser. Just Google UCSC genome browser or go to genome.ucsc.edu. This genome browser was developed as part of the mouse and the human genome reference effort back in the late 90s, early 2000s. The aesthetics are indicative of that. But what's really amazing about it is, you can pull up, you enter your species, and you click on a link, and all of a sudden you've got this fast, interactive, and incredibly informative interface to exploring the genome and all the annotations and basically all knowledge about that species. And up at the top, there's this little interface where you can type in a gene name that you're interested in, or you can type, the convention is to type chromosome, then a colon sign, then the start coordinate dash end coordinate, and that'll jump to a particular position in the genome. So I'm going to just take a quick aside here and go there. Anyone working on a species other than mouse or human that might be in the UCSC genome browser and we can try and look at? Everyone's mouse or human researchers. Okay. Well, Michelle, you sure you're not working on bat genomes? Are you just knowledgeable about bat nomenclature?
Let's do banana slugs for UCSC.
That's, one would hope that the banana slug was actually in the UCSC genome browser. Let's see. I have no idea what the Latin name is for this. Let's see. I think we are out of luck.
I would suggest a bat, but I don't know which one are available.
All right.
There might be a Myotis something.
Let's look through here. I see no bats. Oh, I do. Hey, look, megabat, microbat. So there's like one. Okay, so the little brown bat is representative of the microbat group. Let's go with it. Let's see what's there. So when I clicked on that up here at the top, the shorthand name for the microbat or the little brown bat is Myoluke2. So we're gonna hit go. And all of a sudden, it takes us to some arbitrary location. I actually don't recall how they choose where you start out. But it looks like this assembly is fair. The assembly for this microbat genome is fairly immature. It doesn't have like a CHR chromosome label. It's got some, more cryptic accession number for some sequence in this genome. But the main point that I want to show you holds up is that, here's some, we can zoom, let me try to make this a little bit better. So let me just show you what I did. If you go to configure, you click this configure button, and you can make, I have a huge screen here, so I can make this a little bit wider. And I have bad eyesight, so I'll make it 18 point. I don't like alphabetic fast, so we'll go with times. And you can play with all this. Hit resubmit. You get a bigger image with a different font. But so what we can see is there's some gene annotations. There's some gaps up here, gaps in the reference genome. So these are gaps would be situations where, like we learned very early in the course, where the nucleotide sequence ends for some reason. Sorry, Sarah, we didn't get to seals, but I'll use seals for next year. So we have sign elements, line elements, all these annotations that were computed by repeat masker and these simple repeats. Now, what the annotations for microbat are pretty scarce. There's CPG islands, so we can, down here at the bottom is a list of all the different tracks or annotations that are available for this species. For human, it's completely overwhelming. There's like literally hundreds of them, but we could turn this on and show where CPG islands are. So we go, by default it was hidden, we can click show and then we hit refresh, and then voila, all of a sudden, what we should see, yep, is we got this new track here where CPG islands are. Okay, and you can do lots of different things to configure how much detail they show for each of these annotations. For instance, sign elements. There's so many of them, it just like densely packs them in this like kind of like a rug plot here. But you can, if you want to see all the detail, you can. Oh, sorry. If you want to see more detail about sign elements, you can zoom in here. So if you click your mouse and drag up the top on the scale, you can zoom in. And now we're seeing individual sign elements. And if I hover over it, it'll tell me what type of repeat it is. And if I click on that, I can actually get more information. It'll tell me like more information than I ever want to know about that particular sign element. We don't have enough time in this course to go through all the things that you can do in the UCSC genome browser, but I would say that I would recommend that you spend some time familiarizing yourself with it. There's some training tutorials that they have. It's an incredible resource, and I think it's underappreciated how much you can do with this. You can actually do analyses. You can do alignment with this. You can compare and contrast genomes. You can design PCR primers in the UCSC genome browsers. There's tools here for in silico PCR, so you can figure out how many, whether a primer is degenerate, how many off-target things would it be. You can BLAT is the UCSC genome version's analog to BLAST. It allows you to just pop in a sequence, nucleotide sequence, and it'll tell you all the locations in a particular critter, whether it's microbat or a sealed genome or the human genome, all the locations in which that exists. There's too much to go over, but definitely check out their training. So it can help. There's a bunch of different tutorials, and I think they even now have a YouTube series. But every time I dig into the UCSC genome browser, there's new functionality that I didn't know about, and it's just awesome. Okay, so enough of a sales pitch about UCSC genome browser. What you see graphically when you go to this website, you know, all these annotations are drawn as genome intervals. This is on chromosome GL4299775 and some position X to Y, start to end coding. Often what we want to do is download a file which represents the information that we're observing on the UCSC genome browser, because that's what we're going to need to compare, say, genetic variants to our tools, like, you know, using bed tools, which you'll learn a little bit, or BCF tools, or any of the tools that you would use in R or on the command line in Unix are going to depend upon files. You need to compare this file to this file to figure out all the interesting bits, right? Well, how do you get the files? That's what I want to focus on now. So I'm actually going to ignore the slides for now. I'm just going to go through an example of how to download some data files from UCSC genome browser. So I'm going to pick a different species. I'm going to go now with, let's move back to human. That's what I know best. The latest build of the human genome is HG38. I want to point out what I was talking about HG19. So the last build of the previous build of the human genome is called build 37, or GRC, which is the Genome Reference Consortium, H for human, build 37. So why is it also called HG19? Well, the Genome Reference Consortium is sort of the master record of all the different versions of each species genome. It had version 38, but not all of those 38 versions ended up in UCSC. So UCSC had their own numbering system, and it led to tons of confusion, because even back in 2003, UCSC was on 16. It was called build 34 elsewhere. In 2013, with the release of build 38 of the human genome, UCSC agreed to change their numbering scheme to skip from 19 to 38 just to synchronize everything and make things less insane. Okay, so I can go to the UCSC genome browser for human, And I like this gene called ATM, which is a kinase that that phosphorylates different downstream proteins in a response to DNA damage repair. It's this massive gene, lots of introns and exons. But check it out. There's all these other annotations. There's conservation across different species. I have all this stuff turned on. There's repeats. There's lots of other stuff. And if you scroll down, you can see how many more potential annotations there are for human versus brown bat. Okay, so there is a tool within the UCSC genome browser that is absolutely amazing. It's called the table browser. And what the table browser is, well, the table browser is a database of all the chromosome start and end coordinates of all the things that you could possibly see on any one of the species genome browser views. You can actually download all the chromosome start and end coordinates for all these different annotations, not only for this locus, but for the whole genome. So in short, the table browser is how you download files that you would want to use for analysis. So if you're following along at home, go to Tools, Table Browser, click that. Okay, and now we get this very 1990s website, but trust me, it's worth suffering through the aesthetic limitations. It's pretty amazing. So it defaults to you pick what clade you're dealing with, and then what species. So for mammals, there's all these different species that you can get data for. If you're into pig, panda, microbat, megabat, marmoset, whatever, we're dealing with human. And then once you choose that species, you have to choose what genome build you want. Now this is critical because with each version of the human genome, the chromosome, the sequence changes. They might add a few nucleotides or delete a few nucleotides if they refine what the sequence is. Every time you add or delete nucleotides, that changes the coordinate system. So the chromosome start and end coordinates that you have for a particular annotation are always with respect to a particular genome build. Genome coordinates for one genome build do not apply to genome coordinates from another genome build. So this is a common problem in genomics or bioinformatics where you've got like genetic variant coordinates in one genome build, say build 38, and then annotations for a different genome build. That's a no-no. You need to make sure that all your analyses and results files and your annotations that you're trying to compare and contrast with are on the same genome build so that the coordinates match up. So we're dealing with build 38. Then there's a whole litany of different types of annotations that we can get. It defaults to genes and gene predictions because that's what most people want to look at. I'm actually going to go to let's see, I'm going to do regulation, which is for gene regulation. And I'm going to do CPG islands. So this, I'm going to show you how I got the CPG island track that we used like 4 homeworks ago. So when we choose that track, what it shows us behind the scenes is the database table name that is stored at the UCSC genome browser that stores all the chromosome start and end coordinates for the CPG islands. So I can click this button. It's a bit jargony. Describe table schema just means tell me what the columns are in that database table. And what you can see is the 3 most relevant things here are the chromosome, the start, and the end coordinate. And you might be able to squint at this and see, like, here's an example, gives you sample rows from this database, chromosome, start coordinate, end coordinate, and then name. Now, if you remember, the bed file that we used for CPG coordinates were essentially just these 4 columns, right? And I think I might have done something to get rid of this white space and put an underscore there or something. But the main point that I'm trying to show here is that all this information is stored in conceptually a spreadsheet, also known as a database table, on a server in Santa Cruz, California. And what's so cool about the table browser is you have this little interface to get data out of that database in Santa Cruz and put it on your computer for analysis. All right, so I'm going to skip. There's a lot of other options for things you can do with these database tables. We don't have time to go through all of them. The tutorials that I mentioned will take you through them. But the most common use case is to download the data that's in this database table for the CPG Island track in different formats. So you can say, all right, I want to download it in bed format. If I don't give it a file name, if I just leave it empty and click get output, this is an intermediate thing where you just almost always say click get bed here. And what it'll show you is the data that would be downloaded. Now, why is it just this one coordinate on chromosome 11? Well, if I go back, that's because in this region interface, I left it as position only. So it's only giving me the CPG islands within this interval here. That's defined by, let's see, chromosome 11, start coordinate, dash end coordinate. Well, if I want the data genome wide, I need to click genome. Now, if I click get output again, click get bed, now I just get page after page. It'll probably break my browser because it's vomiting all the data from that database table to my screen. Okay. This is just so you can see the type, what the data, it's so you can sort of sanity check the data that you're downloading. Does this look the way I want it to? It's not very useful just as a big vomit of text on your web browser. What is more useful is if I give it a file name, and I just call this cpg.ucsc.bed. Now, when I click get output and get bed, for me, it's going to pop up an interface where you can see my desktop where I've got my U-Haul order and my kid's Minecraft, but I can click save. And now it's automatically downloading this bed file onto my desktop. So now if I go to my desktop, I should be able to use head. This is not downloading, obviously, to Malibu. This is to my local machine. If I wanted to then put it onto Malibu, I'd have to use SCP to put it up there. So I'm just working on my local machine right now. But I can just do that. And yeah, look, it's just like what we saw. This is exactly what I downloaded. This is exactly how I downloaded the file that I used for the first or second homework. Okay. Right. So the other thing I wanted to show you is when you choose bed format, it pre-selects 4 columns for you. Obviously, it picks out chromosome, start, and end coordinate, and then it makes this composite 4th field, which is basically an annotation, a name for each of the CPG islands that it pulled out. You have control over that, though. You can say selected fields. This is, again, a bit jargony. Selected fields, which means columns, from primary and related tables. Primary means the table that we're dealing with, CPG Islands table in the UCSC database. And then there's other related tables that you might be able to get extra information from. So if we click that and click get output, now it's going to give us another interface where we can actually get more information if we wanted. So let's say I want chromosome, start, and end coordinate. That's the interval in this reference genome coordinate system. Let's say we also I want that CPG island name, just like I already have. That's this bit. But now I want more information. I would like to know the length of the CPG island. I'd like to know how many CPGs there were in that island. I'd like to say, get the GC number. So as I forget who proposed this a while ago, but GC content is the number of C's and G's divided by the length. Well, we got the number of CPGs here, we got the length. So now we can we're going to compute GC content. Well, maybe we shouldn't, because they actually have a column for that. So let's just grab those as well. So now when we click get output, instead of 4 columns, 4 fields of output, we're going to get 1, 2, 3, 4, 9. Okay. So I am going to call this cpg.ucsc.extended. Let's take a look at that. So now, what's really nice about the UCSC genome browser in this case, it gave us a header line. The first line, it starts with a # and tells us the name of each of the chromosomes. This is super nice because if you remember BioLock, now we've got column names where we can actually refer to these columns by name. But as you can see, I'm going to use, so if you pipe data to column, a Unix command called column-t, it will align, should align. Second. Well, it mostly aligned the column names with the data. It got a little messed up because there's an extra space here in the CPG island stuff. But it's showing us the GC content and the number of the length and the number of GC G's and C's in the annotation. Point of all this being that UCSC table browser is really flexible. You can get access to all this really useful information. So you could download this and you'd be like, all right, what is the longest CPG island in the bat genome and how many G's and C's are in it? You could just download this, use your aux skills and find it straight away. Or even just sort the file by GC content. The last thing I want to say about the table browser is, there's just tons and tons of different annotations that you can get. So you can go, if we go back to genes and gene predictions, there's tons of different gene predictions for the human genome. So we'll get all these different versions of protein coding genes that are predicted by something called GenCode and then RefSeq. They're basically different projects for predicting and annotating where genes are and are not. You can get protein domains in genome coordinates. You can figure out where tRNAs are, et cetera, et cetera. All of this is accessible. So let me just show you. I'll switch to PFAM domains. All of a sudden, it's going to switch the database table that's relevant. And I can click the database, describe database table. And we can see that now there's information about where these 7 transmembrane domains are and G protein couple receptors, or I don't know what a SAM domain or a SAN domain is, but you can get all that information. So in a nutshell, the UCSC genome browser and the table browser are essentially your clearinghouse for getting all these annotations that are constantly created and refined both computationally and molecularly within each of the different genome research communities. So I have left slides for posterity. So if this went too quickly or you don't want to watch the video again, all these slides show you how to select different gene or other annotations, save it as an output file, and look at the content. So you should be able to do this for your own species that you're working with, or whatever annotation you want. I also wanted to include some slides about an equivalent resource from a European effort called Ensemble. You've probably used Ensemble before. The website is a little more complicated to use, I think, than the UCSC Genome Browser, but it also has something called Biomart. Let me just show you the right slide here. So if you select Biomart on the Ensemble Genome Browser, so let me just show you. So it's Ensemble without an E at the end. Click Biomart, and similarly, you choose what type of database you want. Let's say we want to get Ensemble gene annotations. You then have to choose your species, tons and tons of different species here. I saw Chinook on here, so let's pick salmon. And what we get here is an interface where we can choose different types of attributes about salmon genes that we want to pull out. So we might want to know the chromosome that the gene is on, and the gene start, the gene end, and the strand. In Biomart, we can then go to this results tab, and it will show us automatically example output based upon the attributes that we selected. So we've selected the gene stable ID, the gene version, the transcript stable ID and version, and then chromosome, start, end, and strand. So one difference here is that Ensemble denotes the forward strand as a 1. UCSC convention is to have a plus sign. Ensemble uses a -1 for the negative strand. UCSC uses the minus sign. So again, there's some inconsistency in genomics and in bioinformatics in general. It's something we just have to get used to, unfortunately. So assuming that we feel comfortable that these are the attributes that we want in our file, we can then download the file as a TSV file. You could make it HTML if you wanted. I don't know why you'd want to. More common use case would be to download it as an Excel file right away. But I am going to download it as a TSV file. TSV stands for tab separated values, or tab delimited text file. We click go. This data is coming across the Atlantic. So we get an export file. Let's see what that looks like. So now this is exactly the data we were looking at on the web page, but in a much more useful tab delimited text file that we now know how to work with awk and grep and sort and cut and whatever. But just to wrap up here, what I want to show you is that you can also filter the information that you get back from Ensemble Biomart by lots of different things. You can, again, I could just limit it to chromosome 4. It'll download just the annotations relative to that chromosome. I can filter things based upon a particular range. So I could say chromosome 13 between position 100 and 200, for instance. You can filter, if you just want annotations for a particular gene ID, you can put them in this interface and it'll limit it. So essentially, the Biomart is really just a query builder. You're building up a request for a certain type of information, and you may want to subset it in different ways or choose different attributes about it. Again, there's so much that you can do with Biomart that it's well beyond what I can really cover in even 1 or 2 lectures. But what's really nice is just like UCSC, they've done tremendous work putting together tutorials about how to use, there's videos about how to use Ensemble in general, and then there's really nice videos right down here about how to use, to get annotations with Biomart. So if this is something that you will be doing in the future, I recommend watching these videos, drink it, watch it in the morning with a cup of coffee or something. But also just remember that if you forget how to do this, come back to these resources.
There's also an R package for Biomart.
Oh, that's right. Yeah.
Um,
Do you know
slightly.
what that is?
What's that?
What do you know the name of it?
It's just Biomart with a capital R.
All right. Yeah. So
It's a bit excruciating to use at first because the syntax is quite confusing. But then you have a like programmatic record of what you downloaded, which is useful.
Yep. So yeah, Biomart R package, and I don't think UCSC has anything equivalent to that. But the main point of this lecture is that a lot of times you know exactly what you want to do, what type of analysis. You got your RNA-seq data or you got your SNP calls. The challenge for a lot of people is figuring out how do you get the right annotations to ask a question that is already at the tip of your tongue. And really, the UCSC table browser and Biomart are the clearinghouses to get that type of information. And hopefully by now, you at least are somewhat comfortable with Unix and potentially R to download those files, load them as a data frame, use awk or other tools that we're going to learn over the next few lectures to compare and contrast your experimental data with the annotations. But the hardest part sometimes is just knowing where to get the annotations. So next lecture on Thursday, we're going to talk about some of the file formats that are used for storing these annotations and suffer through some of their idiosyncrasies in prep for next week of actually using those annotations to do what I hope will be some fun and somewhat cool analyses. So with that, any questions before we wrap up? Okay, well, I will see you all on Thursday for a discussion about genome annotation formats. See you then.
Thank you.